chemical control? I sweat and shake, hear me moan, oh no, my hormones! All right, so again, although Western medicine has made so many contributions to health, it was the Chinese, an Asian source, that made the first real documented strides. So over 2,000 years ago, Chinese medicine, the Chinese apothecary, had identified various pituitary and other androgenic hormones isolated from the urine. They had isolated it from the urine and also, in fact, from semen. I didn't include that there. But they use, and this is the text describing that, and they use these for the treatment of various ailments. So they have known for a very long time that there were some sort of chemical agent that had a profound regulatory effect on different physiological manifestations. All right? So for several thousand years now, a couple thousand years, that has been known in China. In Europe, it took till the 19th century, 2,000 years later, for Arnold Berthold to put this together, that castrated cockerels or castrated roosters, we call them cockerels, they did not, so here's a regular untreated rooster, and if you castrate them, they don't develop the gender-associated traits of the rooster. So their combs and wattles are significantly reduced, and they did not show that overtly male behavior, the crowing, the dominance, all that was absent. And he was at the University of Göttingen. Am I saying that right? Göttingen. All right, so there's that, right? That's the only history I'm giving you there. Here are the tissues associated with the endocrine system. They're many and varied, distributed throughout the body. So this is just kind of an introduction to the characters that are at play. Because this is a chemical control system, there is not a single centralized tissue that encapsulates this system. Although there are a couple, there is a bit of a hierarchy, and there are a couple at the top of that hierarchy, the hypothalamus and the pituitary gland being the ones I had in mind. So the hypothalamus is part of what's called the diencephalon, if you'll remember back to my CNS lecture. And the diencephalon is composed of the thalamus and the hypothalamus, part of on the upper end of the brainstem, sort of the junction between the brainstem and the cortex, the cerebral cortex. The thalamus is where it's a major relay station for projections up into the cortex for conscious awareness. The hypothalamus is intimately associated with that, as the name would have you understand, and it descends from it, and then is the source of ADH, the antidiuretic hormone, which we talked about in the last unit, oxytocin, which I think I talked about when I was talking about positive feedback and childbirth. Did I mention oxytocin during that? And then other regulatory hormones. This plays into the pituitary gland. So the pituitary gland is this little berry-shaped gland that is on the very bottom of the inferior surface of the brain. It's divided into two portions. There's the adenohypophysis and the neurohypophysis. So the pituitary gland, another name for it is the hypophysis. And there are two portions. The anterior lobe is the adeno hypophysis. You can remember that because anterior and adeno both start with A. And then the neurohypophysis is the posterior lobe, and N is later in the alphabet than A, as is P. So the posterior lobe is the neurohypophysis. Anterior lobe is an endocrine sensitive. It's a target. It's a target organ for hormones that are released by the hypothalamus. A whole host of them there. So various stimulating hormones, growth hormones are produced there. Prolactin, follicle stimulating hormone, luteinizing hormone, etc. And then the posterior lobe, the neurohypophysis, actually gets neuronal projections from the hypothalamus. We'll see this in detail, but this is where the production of oxytocin and ADH happens in the posterior or neurohypophysis. Some other organs will stay in the brain. The pineal gland, the sort of third eye of the brain. Does this help if I do this? I love the pituitary gland. This is where tryptophan is converted to serotonin, which is then converted to melatonin. Melatonin is, that over-the-counter drug that travelers sometimes buy to help regulate their sleep cycle. There are actually photoreceptors in the pineal gland. However, it is a non-image forming organ. You don't actually form an image with the pineal gland, as I'm sure you're all aware. However, it is ancestrally related to what's called the pineal eye. For example, in iguanas. Has anyone ever hung out with an iguana before? Well, for any of you who do get a chance to hang out with an iguana, if you look on the surface of an iguana's head, there's a little dark spot kind of where your apple core would be in your hairline. And that is, our pineal gland is the inheritor of what is called the pineal eye in those lizards, because it regulates their circadian cycle. It is light reactive to what time of day it is, etc. And our pineal gland has similar functions. However, it is not really exposed into light and dark the way theirs is. Then descending into the neck, we have the thyroid and parathyroid glands, which we've talked about a little bit. If I get time to, on Wednesday, half of my lecture will be about the thyroid gland, although I kind of doubt that I'm going to get through much of that. We'll see. I've certainly talked about the parathyroid gland with you in calcium homeostasis. Then the thymus gland. So the thymus gland, did I talk about thymic deletion in here yet? Thymic deletion, the immune system, and like the first week postpartum of birth? No? Well, the thymus gland is a pretty interesting gland, and most of you at this point in your life, certainly in me, the thymus gland has atrophied, and there's not much of it left. But this is a gland of childhood, and most importantly, that first week of life. What happens is that the thymus is involved in the process called thymic deletion, whereby T cells that are produced by the baby. So when your baby's born and it has the mother's immunity, right? And during that process, I'm going through this now because I'm not going to be able to go through them in more detail later on. During the first few days of life, T cells that are self-reactive, T cells that recognize your own muscle cells, your own liver cells, et cetera, these self-reactive T cells are they're turned on, and then they pass through the thymus and are deleted from the population. They're killed. And so that with time, you are not, any T cell that it recognizes the self has been turned off and deleted from your population, so that your immune system is only geared to recognize things that are not self. Yeah.
So sorry, at what point in the life cycle does this happen?
Postpartum. Yeah.
In the mother.
No, in the child. Yeah. The baby's born, and all of their self-reactive T cells are deleted from the population in a process called the thymic deletion. Thymic deletion by the thymus. So that's just a little of the background on what the thymus is. The heart. I didn't really get to the heart. Produces the natriuretic peptides the right time, so the atrial natriuretic in the brain, or B-type, also called B-type natriuretic peptide. I had one slide in the last unit where they appeared, but it's kind of the other side of the homeostatic coin from the RAA axis that we talked about. I won't get a chance to go into that in any more depth at all. The suprarenal glands, Mr. Laser's favorite gland, has a medulla, which is responsible for the fight or flight hormones of epinephrine or norepinephrine, also called adrenaline or noradrenaline previously, and the cortex, which is where we get the cortisol, various corticoids, aldosterone, and various androgens are produced there in the cortex. Kidney, we talked about. Your fat tissue, actually, so that's pointing, that arrow here is pointing to the omentum, which is the fatty skirt that hangs over your intestines. It's been cut there a little bit, but fat is where we produce leptin, also ghrelin. These are hormones that regulate appetite. So whether you feel full or hungry is going to be regulated by hormones that are produced in the adipose tissue. Sadly, we probably won't get a chance to talk about those too much. There's a host of hormones in the digestive tract that primarily act and live within the digestive tract. The pancreas. So there are endocrine and exocrine cells in the pancreas. The exocrine cells we will talk about during the GI lecture in the latter half of the week here. But the endocrine cells produce insulin and glucagon. Okay, and I will hopefully get a chance. I'll make sure to talk a little bit about the function of that, so you'll have some basic background for your insulin receptor talk. Just where's the insulin coming from, kind of thing. And then the gonads. So in males, these are the testes, and the ovaries in females. These are making various androgens. The male, it's primarily testosterone inhibin, and then estrogen, progestins, and also inhibin in the ovaries. We will probably not get much of a chance to talk about those. All right, so to review the types of intercellular communication that we've talked about this year. All right, so how does one cell keep in touch with another? There is direct communication via gap junctions. I talked about how exciting those things are to me when I first introduced them. So this enables a sort of synstitial arrangement where small solutes, ions, whatever, can pass from one cytoplasm to the next. And so some examples of that, there's going to be neurons that use gap junctions. We know cardiac muscle cells use gap junctions to promote the synstitial arrangement. And then there's the paracrine communication. So paracrine a paracrine cell is one that is going to degranulate or release its vesicles, and that stuff is going to get dumped into whatever extracellular fluid surrounds that cell, and it's going to be a mixed cell type tissue where there's some other cell there that is responsive to that hormone that has been released into the interstitial environment that those cells find themselves in. So directly changing the milieu of the cells in that tissue without involving the circulatory system. Does that make sense? Lindsay, you're just, okay. Any questions on those two? All right, so then I should probably switch these. Synaptic communication we talked about at great length, actually. We discussed how action potentials change the membrane potential dynamics and then cause a calcium-mediated degranulation of these vesicles that have whatever neurotransmitter it may be, right? So there can be acetylcholine or whatever, all the different, the glutamate, et cetera, that are going to attach to some sort of chemically gated channel on the other side of the postsynaptic membrane and the downstream neuron or potentially muscle cell or whatever effector it is. And then finally, we have here this endocrine communication. And this is mediated through the circulatory system. So these cells, endocrine cells, are going to produce their secretion, whatever they're intended to make, whatever hormone or what have you that they're making, is being released into the lumen of some sort of vasculature. And this gets pumped throughout the circulatory system, and this tends to be much more systemically or widely distributed in terms of the communication. These hormones can have multiple target organs this way, so it can coordinate a rather sophisticated response instead of like a very discrete targeted response, for example, in some kind of paracrine. All right. Ooh, look at that slide. So we're going to walk through this slide one little bit at a time. And we're going to talk about the types of hormones that there are. And you can classify these in two ways. First of all, what is the chemical structure that they have? And then how do they act? What mechanism are these hormones using to effect the change in the target tissue that they are designed for? All right. So let's just look at an overview of the structural classifications first. So there's three broad categories. There are either amino acid derivatives. These are small molecules that are some sort of modified amino acid, some sort of derivative of an amino acid. Then we can take a series of amino acids, a small chain, and make these peptide hormones. All right, so small chains of amino acids are called peptides, and certain peptide chains have hormone and endocrine activity. All right, and then the third category is these lipid derivatives. So fats of some sort, fats or oils in your body, can be derivatized into some kind of, again, usually small compound that has some sort of endocrine activity. And these peptide hormones are usually the biggest molecular weight, the largest molecular weight category of them. So, all right, let me see here. There are two categories, mechanistic categories. There is this extracellular mode of action where a hormone is going to bind to some sort of receptor on the surface of the cell, and that's going to cause a cascade of second messengers. All right, so some other messenger other than the original hormone that was circulating in the blood is going to cause a cascade in the cell. And I have here a G protein coupled receptor. This is not the only way this is just an example of one of the extracellular modes of endocrine signaling. All right, but just to show the example that you have a hormone that's going to bind to some membrane-embedded receptor that's transmembrane, and that signaling is going to be transduced by some conformational shift in this transmembrane protein that's going to have a bunch of downstream effects. Okay, and in this one we have a G protein. This is a protein that's coupled to a guanosine nucleotide. That this protein becomes activated once the hormone binds, and it can have effects on cyclic AMP or effects on intracellular calcium, which then becomes what's called the second messenger. So cyclic AMP, in this case, or calcium become the second messengers. They are the proxy for whatever hormone this is. All right, the hormone is carrying a message, and then that message gets transduced, and inside the cell, it's the cyclic AMP or the calcium that is going to continue to propagate that. That's the second messenger system. Many of you have probably already heard of this, I'm guessing. So then there's also intracellular signaling. And this happens when the hormone is able to pass directly into the cell. It's able to pass straight through the membrane and come into the cell. It's probably not a surprise that most of these are going to be, in fact, all of these are going to be fairly hydrophobic or have hydrophobic components to them. Often they're amphiphilic, meaning there's a hydrophilic portion to them. But they're able to solubilize through this fatty portion of the membrane, this non-polar port of the membrane. They're able to come right through it, and they then directly signal within the cell in a host of ways that we'll see in the abstract in a moment. Yeah?
Are all lipid hormones pretty much completely soluble in the blood, or do they kind of form micelles, or what?
Yeah, in terms of transport of lipid hormones, there's a variety of ways that they can be transported, often protein associated, with like albumins or whatever, maybe some globulins as well. But there's a lot of different transport mechanisms for them. Some of them are directly soluble. All right. So the first we're going to look at is a little more detail are these G protein signaling. Has anybody had this before? This stuff before? A few of you? Just show of hands. Like half the class has had this stuff before. All right. And the category, what I've done over here is shown the categories of cells, or hormones, pardon me, that utilize this general mechanism, okay? So we have the amino acid derivatives, one of them being tryptophan. It's a derivative of, this is melatonin. I mentioned that melatonin was produced by the pineal gland and regulates the sleep cycle, stimulates dreaming, etc. And then the various catecholamines that we've talked about a couple times this semester, such as epinephrine and norepinephrine, the fight or flight hormones, dopamine, everybody's favorite hormone. So, and then we have these peptide hormones. First of all, there are the short polypeptide and small proteins. These are hormones that are under 200 amino acids in length, so relatively small, actually, all things considered, up to these glycoproteins. So these glycoproteins can be actually fairly high molecular weight. So, for example, there's TSH, the thyroid stimulating hormone, and luteinizing hormone, and follicle stimulating hormone that are important in the menstrual cycle. Erythropoietin that we talked about a little bit, the hormone for producing red blood cells, is a fairly large glycoprotein, and then inhibin in reproductive organs. And then these short polypeptides, there's a long list of them there. yeah, I'm not expecting you to memorize that list at all. But how does this work? So mechanistically, there is going to be the binding of the hormone to some sort of transmembrane receptor that is coupled, a Gprotein coupled receptor. That Gprotein, this protein that incorporates the guanosine nucleotide, is inactive until the hormone binds. It becomes activated, and depending on the cell type and the hormone pathway that we're talking about, one of two type mechanisms begins to get engaged. The first here is this one that uses epinephrine, norepinephrine, calcitonin, PTH, the antidiuretic hormone, some of these regulatory ones, glucagon. It activates adenylate cyclase. So what adenylate cyclase does is it hydrolyzes ATP and then also cyclizes it. It hydrolyzes it to AMP, uses that energy to also form a cyclic bond, and we form cyclic AMP. Cyclic AMP is now the second messenger, and that's going to have a whole host of downstream actions. It's going to turn on various kinases. What's a kinase? Anyone want to share what a kinase is? Anyone know what a kinase is? Yeah, that's right. It adds a phosphate group to something. And typically adding a phosphate group, not always, in fact there's probably thousands of examples of the opposite, but when you add a phosphate group that's often associated with activating that enzyme or whatever, that member of a signaling cascade. So you put the phosphate on it, it's now active, and it goes on to do its business. So these kinases can do all kinds of things. They're able to open ion channels, perhaps, if that's what the cell is designed to do, or activate various enzymes, whatever. There's a lot of downstream things that happen. The other generalized pathway that uses these Gprotein coupled receptors, the activated Gprotein is going to turn on phosphodiesterase, and phosphodiesterase is going to turn this cycle off. So it's going to hydrolyze the cyclic AMP to AMP, and this AMP is going to lead to a host of other downregulation of a lot of the terminal effects of the adenylate cyclase pathway. So on the right hand is how this pathway is turned. Cyclic AMP pathways can get turned on, and this is how they can get turned off. All right. So by the production of AMP here, this release of AMP, this will, for the people who are talking about AMPK, you will recognize that is going to shift the AMP ATP ratio in the cell. I don't know how far into the material that group has gotten yet, but that is going to have an effect on downstream AMPK signaling. All right, and that's mediated by phosphodiesterase. This is a simplification of this pathway. There's a whole host of intermediaries and accessory proteins involved in that. All right. So next one. The next type of extracellular signaling. We can have calcium act as the messenger, and let's see how this works here. The hormones involved are also going to be, the catecholamines can again be involved in these kind of pathways. You'll find some short polypeptides that work this way. And then our first lipid-derived hormones to enter the story, these eicosanoids. And so they're all derivatives of arachidonic acid. So there's a number of categories of eicosanoids. Eicosanoid refers to eicosanoic acid or arachidonic acid. Eicos means fish. A lot of times a lot of them are derived from fish oils. So the leukotrienes, which in general are pro-inflammatory, the prostaglandins, which often are anti-inflammatory, thromboxanes, prostacyclins, just categories we won't even touch other than mentioning that they exist. Here's an example of what prostaglandin E is going to look like. And you can see how this is going to be a derivative of a fatty acid. So here's that carboxylic acid moiety on the end, and it has been oxidized halfway down the chain and cyclized as well. All right, so these are the characters, the types of hormones that are going to have this type of signaling. How does it work? Hormone binds to the protein, the extracellular receptor, activates the Gprotein. It's a Gprotein coupled receptor, and that is going to activate a membrane-associated protein called phospholipase C. Phospholipase C. And phospholipase C is going to have hydrolysis of a triglyceride, and you're going to get diacylglycerol first of all, that is going to activate protein kinase C, PKC. When protein kinase C gets activated via this small molecule diacylglyceride, it opens a calcium channel, and the calcium is able to flood into the cell. When that happens, calcium does now become a second messenger. All right. So rather than having cyclic AMP, our second messenger is now calcium, and calcium is not being necessarily sequestered by the cell, but it can be sequestered from internal or external stores. So if it's being released by internal stores, that's getting triggered by the other hydrolysis product of the triglyceride, the inositol triphosphate. So I drew those compounds. I showed that reaction on the board that one day. I drew those molecules and showed the hydrolysis reaction for you guys, if you throw your mind back. But anyways, once this happens, once we have calcium either from sequestered internal vesicles or from extracellular influx, then this calcium binds to calmodulin, and calmodulin is going to be have an effect on the downstream signaling. All right, so that's going to start the signaling cascade that can go into a host of different directions depending upon the cell type and the hormone that we're specifically talking about. These are just very general overviews of the function. You can begin to appreciate how massive the discussion of the endocrine system is. Has an enormous number of permutations. All right, so let's switch now that we've covered all the extracellular mechanisms of the endocrine system. Let's talk about some of the mechanisms it uses for intracellular signaling. And the first is going to be the steroid hormones. And these are hormones that are mostly derived from cholesterol. They're mostly derived from cholesterol. So in the gonads, this is where we get the androgens, the estrogens, progestins. In the suprarenal cortex, we're going to get the various glucocorticoids, the mineralocorticoids, and there's also some androgens that are produced there that I had mentioned earlier. Calcitriol is one of these steroid hormones that if you, maybe our first or second lecture, if you throw your mind back, I talked about that. All right, so and here's an example, estradiol, or it's a member, the most famous member of the estrogen family. Okay, so how do they work? How do they work? A lot of them have this structure. So I said they're structurally related to cholesterol, and so they have this polycyclic structure that you see depicted here. Very hydrophobic, typically hydrophobic compounds. First thing that happens is they just slide on through the membrane. They just slide right through. They are able to dissolve into the fatty lipid bilayer and are now inside the cytoplasm. So what happens now? So they can bind to a number of target proteins, all right, and it depends on the cell. This can either be in the cytoplasm, or they can translocate directly to the nucleus. They pass through the nuclear pore complex here, and are able to go pass go without going to prison here, and they're going to bind to some sort of receptor that's going to be a transcription factor in a binding in a complex that's bound to DNA. So you're going to have DNA that has potential to be transcribed. This hormone is going to bind to a receptor that's part of a transcription factor complex, and that's going to lead to downstream gene activation. That gene is going to turn on. It's going to be transcribed and then translated. So transcribed into messenger RNA. That messenger RNA is going to get translated into proteins. So we'll have some sort of protein synthesis that's going to have whatever effect that protein has. Okay, am I in the way here? I don't know. I go by the watch on the wall. That other class seems to get out early to me. All right. So then the thyroid hormones. I'm actually right on time. This is just about right. The thyroid hormones are, here's a picture of T4 thyroxane. 4 is called, the subscript 4 refers to the fact that it has 4 iodines incorporated into its molecular structure. There's T3, which has 3, and then there's T4. Most of you are probably aware of the importance of iodine in the diet to prevent what's called goiter or hypertrophic thyroid gland, a large thyroid gland. So similarly, this thyroid hormone is able to pass right through the lipid bilayer, come into the cytoplasm, and it's going to bind either some sort of receptor at the mitochondria. So you're all familiar with the fact that you have the DNA that you have from half of your mom and half of your dad in the nucleus, and then there's your mitochondrial DNA that you have just from your mom. Right? All your mitochondrial DNA comes from your mom. Thyroid hormones can have targets in the mitochondria. This typically leads to an increased production of ATP, and whatever the cell needs the ATP for, that downstream thing is going to happen. Otherwise, it comes to the cell and has a pretty similar abstract pathway, a generalized pathway to the steroid hormones. So it's going to bind to some sort of transcription factor, cause gene activation, transcription, and then eventually translation, and then whatever the downstream effect of that protein is. All right? So I think, yeah, that's it. We're going to stop right there. That is the mechanistic and structural overview of how your endocrine system works. Next lecture, I'm going to get into a few details, just an overview, a little bit of an overview of some of the details of the system, and I'm particularly going to try to focus on giving you guys as much background as I can that will set you up well for the insulin receptor AMPK talks. Okay? Everybody happy, good mood, sunny day. Make sure to take some time to go walk around in it.